I'm Elliot Meyerowitz from the Division of Biology and Biological Engineering at the California Institute of Technology. In the first part, part one, I talked about why it's so important to study plants, because plants are important to humans in a number of different and fundamental ways. And I talked about an approach that we had developed to capture dynamic information from developing plants so that we could see at the same time patterns of cell division, patterns of cell enlargement and movement, patterns of gene expression, and the movement of proteins within individual cells while the plant is developing. And I discussed a particular problem to which we've applied these methods, which is the problem of phyllotaxis. This shows the top of an Arabidopsis plant, and right in the middle is the shoot apical meristem, the collection of stem cells that forms in the embryo and produces the stem, and then the leaves and the flowers of the plant as the shoot grows out from the plant during the course of its life. Around the meristem in this picture we see developing flowers, with the larger ones being ones that form first and the smaller ones all the way to the very small near the center being ones that are forming at the time we took the picture. The picture is taken with a laser scanning confocal microscope, and we've made the plant fluorescent so it could be seen in that type of microscope by having an expression of a particular gene shown in green and the fluorescence of chloroplast shown in red. What we can see in this view is the regular pattern in which the organs, that is the flowers, are formed around the shoot apical meristem, and it's this that we want to understand and for which I'll develop a model. Now here's the living view as the cells develop and as the flowers form, which we talked about in part one. So we can see, for example, at the top here, a flower forming where it begins as a primordium, and then as a result of more rapid cell divisions in the flower, then in the meristem, this flower primordium projects out eventually several days later, creating the sepals, the petals, the stamens, and the carpels that are the floral organs. So we have this live view based on our confocal microscope methods, which enable us to see in 3 dimensions what's going on in this tissue as it develops. What I'm showing here is a similar meristem in which we're going from the top of the meristem down into its depths by changing the focal plane of our microscope, which is what a confocal microscope does. So we're seeing the cells in the top layer, then the next layer of cells, then the next layer, and the next layer. And we can feed this type of information into our computers and do image processing such that we can identify the position, the size, and the divisions of each cell in the shoot apical meristem, what's called in image processing a 3-dimensional segmentation. And this was done with software created by colleagues of ours in France, the laboratory of Christophe Gaudin. We can see each cell and follow in great detail what's happening as the flowers develop around the shoot apical meristem. So our question is, how do we understand how it is that the shoot apical meristem, this collection of plant cells, makes a new flower primordium every 130 or 140 degrees around the meristem, and we use these methods of live imaging to answer this question. Now there's some background that's necessary before we can get to the experiments, because a lot was already known about phyllotactic pattern before we began our experiments. And much of what was known has to do with a plant hormone called Auxin, which chemically is indole 3-acetic acid. It's related to tryptophan, and the structure is shown on this slide. Auxin has a number of different effects in plants. In fact, it was identified in 1926, and the fact that such a substance must exist was pointed out as long ago as 1880. So there's a lot of history to the experimentation with Auxin, and we know a lot about what it does in plants. One thing it does is it induces the new leaves and flowers in the shoot apical meristem. And this was shown most simply by Robin and Mary Snow in the 1930s, who took little bits of Auxin mixed with lanolin, sheep grease, and they dabbed it onto meristems, and wherever they dabbed it, if it was around the flanks of the meristem, that invoked the formation of a new leaf or a floral primordium. So we know, first of all, that Auxin is a chemical that causes a new primordium to form, and therefore our understanding of the phyllotaxis pattern becomes an understanding of how auxin gets to the right place in the shoot apical meristem so that it can invoke the development of a new flower. Now, auxin does two things on individual cells when it gets to them. The first thing, which is shown last here, is that it causes changes in gene activity by interacting with a nuclear receptor which reads out in the transcription of new genes that were previously untranscribed in the nucleus of the plant. So that it causes rapidly changes in gene activity when auxin goes onto a cell. One of the effects of these changes in gene activity is that the cell wall is weakened because there are proteins that acidify the cell wall by pumping protons through the plasma membrane from inside the cell to the cell wall region outside the cell. And this causes the cell wall to weaken and therefore causes the cell to expand because the cells in a plant are under high turgor pressure. They're all under pressure, and that pressure is restrained by the cellulosic cell wall that surrounds each one of the cells. The pressure can be very high as in a high-pressure bicycle tire, or several times the amount of pressure you would find in a bottle of champagne. And so these cells are strongly pushing outwards, and the walls around them are restraining that movement, which will become part of our story in part 3, not here in part 2. So cell expansion is one thing that's created by auxin, and changes in gene activity are another. And both of these turn out to be involved in how it is that auxin creates a new flower. The position in which auxin creates a new flower has to do with yet another property of this plant hormone, because the hormone has a special system of transport, its own private circulatory system that allows it to move through the plant in patterns that are different from the patterns in which any other substance in the plant moves. That is, the auxin is trapped in each cell, and it's allowed out of the cells by a specific plasma membrane proton, the auxin efflux protein, the auxin efflux carrier, and that auxin efflux carrier is asymmetrically disposed about the cell, so it allows the auxin out only on one side and not on others. And as a result of all the cells doing this in coordination, they move the auxin around the meristem in a particular pattern. We can look at this property in a little bit more detail using this diagram from a review article. So we have here a plant cell, schematically, surrounded by its wall, and this black line is the plasma membrane. Inside the cell, the cytoplasm is at neutral pH, pH 7. Auxin is a weak acid with a pK of around 5, and so that within a pH 7 cell, it's largely dissociated from its proton. It's an ion, a charged molecule. And that charged molecule will not go through the plasma membrane of the cell. So auxin is what's called acid trapped inside the cells. The efflux carrier protein, which in the cells in the Arabidopsis shoot apical meristem is encoded by a gene called pinformed 1, is disposed asymmetrically in the cells. In this example that's shown, the auxin efflux carrier is depicted as green at the bottom of each cell, so that a row of cells in one dimension like this one will have auxin trapped in each individual cell and auxin able to get out of the cell only in those parts of the plasma membrane where the efflux carrier is at high concentration, the bottom of the cell. The auxin then goes out into the cell wall region, and in the cell wall region, the pH is acidic, 5.5, so that the auxin reassociates with its proton and then can freely diffuse either back into the cell of origin or down into the cell below. So we have diffusion acting against transport, and these two processes together cause the net flux of auxin to be from the top to the bottom of a row of cells in which the auxin efflux carrier is found at the bottom of the cells. So auxin has a protein that allows it out of cells, and by that protein not being found uniformly around the plasma membrane of every cell, the auxin can end up being directed in particular patterns in individual tissues. Now, this is a picture of a shoot apical meristem of Arabidopsis taken with our confocal microscope, in which we've made the nuclei green in cells where auxin is at high concentration by transforming in an artificial gene that has an auxin activated promoter attached to a jellyfish green fluorescent protein. And what we can see is the place in the shoot apical meristem where auxin is high are exactly those places where each successive flower primordium will come out, so that we can see 3 primordia in process of forming in the periphery of the shoot apical meristem when we look at the pattern of auxin expression, even though if we were to look at an unstained meristem, those cells would look quite the same as all the others. There's something special about them. They're the sites where the new primordia of flowers are going to appear. Now, if we mutate the PIN1 gene, as was done by Kiyotaka Okada's lab in the 1990s, we don't get floral primordia anymore. So here's a PIN1 mutant, and there's a shoot apical meristem, and no flowers are forming around it. So what does this tell us? It tells us that the way the auxin gets to these particular places in the periphery of the shoot apical meristem is by transport using the PIN1 transporter. And so any model that we have for the angles at which new floral primordia are created has to derive from the behavior of this PIN1 transporter in creating new peaks of auxin concentration. So now we apply our live imaging methods to see what's happening in a real living plant. And what we're seeing here is a fluorescent version of the PIN1 protein in a meristem as it goes over a few days, or a couple of days, and as it's making new primordia. And we can see at this magnification differences in brightness across the shoot apical meristem. Why is this? Because the PIN1 gene and the PIN1 protein are induced by auxin. So anywhere where auxin is high, there's more PIN1 protein. As a consequence, it seems brighter in our live image. So we can see that the pattern in which auxin changes its concentration in the shoot apical meristem is very lively and very dynamic when sped up as much as this has been sped up, several thousand times, and that we get new peaks of auxin forming at places like this where the next primordium will appear, and where the primordia are forming, we see auxin at high levels. But if we look, for example, over here at a primordium forming, it then goes to low level after the flower starts to grow out. And in there lies part of our story, so that we have a very dynamic and active pattern of auxin changes in the shoot apical meristem, and we have to develop a method to understand this so we can understand the origin of the phylotactic pattern. If we look at higher magnification, in a picture like the one I showed before, and when we stain the plasma membranes of the cells red, and we stain the PIN1 protein green, we can see at the boundary between cells, one side is green because the PIN1 is in that cell, and the adjacent cell hasn't PIN1, just the red stain in the plasma membrane. So at each junction between every 2 cells, we can determine from our micrographs the direction in which the auxin is being transported from the cell that has the PIN1 efflux carrier and towards the cell that doesn't have the efflux carrier in that part of its plasma membrane. And if we do that and we draw arrows in the direction in which the auxin is being moved by this efflux carrier, we see that there's a general rule, which is that auxin is always moving up the auxin gradient. Each individual cell somehow assesses the auxin concentration of its neighbors, and it proportionates its PIN1 protein to the plasma membranes adjacent to those neighbors, adjacent to the shared wall with those neighbors, according to those neighbors' auxin concentration. So any cell that has a high concentration of auxin attracts more auxin from its neighbors, and a cell with a low concentration of auxin sends its auxin to its higher auxin neighbors. This is opposite the direction that diffusion would go, and the energy for this is provided by the ATP that creates the pH difference through the proton ATPases in the plasma membrane, so that auxin can move against the diffusion gradient. Now, we therefore know 3 things. One, which is shown first here, is that the auxin efflux carrier moves auxin, and its gene is auxin-induced, as I showed in that brightness changes in the shoot apical meristem movie. This already creates a problem for us to think about what's going on in the shoot apical meristem. Imagine that I'm a cell, and I have lots of auxin in me, so I'm a high auxin cell in any model we might want to make. Perhaps I'm the site of the next flower primordium that's going to form. But because I induce my own auxin efflux carrier, as a result of having high auxin interacting with my own auxin receptor in my nucleus, I send all my auxin out to my neighbors because I have a lot of the efflux carrier. So I was a high auxin cell, and now I'm a low auxin cell. So we have a dynamism there that's hard to think about intuitively. Secondly, since local high auxin concentration causes new primordia, and it gets high locally by transport and diffusion, we have a second problem, which is what's the flux of auxin? What's the pattern of the flux of auxin that would be created by this rule that A, a high auxin cell sends its auxin out rapidly, and B, high auxin cells attract more auxin from their neighbors. And so we can take our three parts to our model that the amount of auxin efflux carrier changes with time relative to the concentration of auxin, that the amount of auxin in an individual cell is equal to the production of auxin minus the degradation of auxin minus the amount of auxin that's pumped out through the efflux carrier plus the amount that diffuses back in, and the direction in which pin 1 sends the auxin is towards the auxin-rich cells. And if we take these differential equations, which represent exactly that English model that I showed before, and we put them into individual bubbles in our shoot apical meristem model in a hexagonal array, and we solve these equations, and where the auxin concentration is high, we make the bubbles look red, and where it's low, we make the bubbles look blue. And then once the equations are solved, we add a new cell in the middle, we do it again and again, and we watch the dynamism in an artificial shoot apical meristem of these equations working the rules of auxin transport, and we get out exactly the spiral phylotactic pattern. So it tells us that these three rules are sufficient to create the spiral phylotactic pattern in a very artificial substrate, which is a hexagonal array of bubbles that look like cells. And in this model, auxin is transmitted instantaneously and automatically in the direction pin 1 points towards each neighboring cell. Okay, you'll notice that we reproduce the pattern of the phyllotaxis of the plants in this simplified computational model, but we do not reproduce the dynamic pattern of auxin changes that we saw in the actual movie. If we look at some stills from that movie I showed before, you can see that the patterns of auxin movement and of pin 1 movement from one side of cells to the other can be very complicated in the shoot apical meristem. We have a primordium over here that's in the process of forming, and all the cells are very high in auxin, as we can see from the high levels of pin 1 reading out the auxin concentration. But after that primordium is established, there's a reversal of pin 1 from pointing towards the primordium in these cells to pointing back towards the meristem that's very lively. It creates a low auxin zone between the primordium and between the rest of the shoot apical meristem, and then eventually the primordium, as we can see here, runs out of auxin, and the auxin is back in the shoot apical meristem and being directed back into the meristem. So you have this complicated reversal of auxin moving first into and then out of primordia that was not reproduced in that model that I showed. And the reason is that we showed instantaneous movement of auxin from one cell to the other without considering what might happen to the auxin in the cell wall that surrounds each of the cells. And the one thing that happens to the auxin in the cell wall is that it can diffuse laterally in the wall into the next cell or back into the cell from which it originated. And if we add that in, which is this set of equations right here, and then we make a more realistic substrate so that we don't just add a cell to the middle of a hexagonal array, but we have cells with 4, 5, 6, and 7 neighbors, just as in a real meristem, and we let all of the cells divide, just as they do in a real meristem, and we apply our new computational model, we get a very close reproduction of exactly what we saw in the movie of the plant forming. And so we've gone one step now from dynamic and live observations of the phylotactic pattern forming as a result of auxin transport in a shoot apical meristem to a computational model based on the rules that we established in our observations. Those rules turned into differential equations that could be solved in each individual cell with a computer. And then in a substrate that resembles a dividing shoot apical meristem, we find out that those rules are sufficient to exactly reproduce what we saw in the real shoot apical meristem. So we have a viable and useful model now for phylotactic pattern. What does it mean to our understanding? And what can it predict? Because I promised predictive models when we began this. This is what this model is telling us, that if we look on the left here, the yellow parts are new primordia forming, and the arrows are the auxin being transported into those new primordia to allow them to be created at those particular positions. Up on the right here is high auxin in the yellow region, and blue indicates a low auxin region because this reversal has occurred at the front of the meristem as a natural consequence of the way auxin is transported in and out of the cells. Why does the reversal occur? Well, because as the auxin is transported toward the primordia, the cells that are transporting it have behind them cells that are not transporting very much because they don't have very much pin1 in them. They're low auxin cells. Eventually, those cells come to have more auxin than the ones in front of them. And so the ones in front of them move their pin1 towards their most auxin-rich neighbor, which is now behind them, and they send the auxin back into the meristem. And the next row of cells does that, and the next row does that. And this reversal is what creates the phylotactic pattern, because each primordium, after it forms, sends its auxin back into the meristem, and where that auxin goes creates the next primordium that will form. This is consistent with an experiment that Ian Sussex did in 1953, in which he took a razor blade and he cut off the forming leaves of ferns. And he cut off the primordia, the earliest formed and later formed primordia, and he showed that if you cut the last two primordia that formed while the next one is forming, it will change its position and the phylotactic pattern will be disrupted. And the reason for that turns out to be that a signal from the primordia, which is auxin, is being sent back to the meristem to create a new auxin peak. And this sending back is a result of the transport of auxin through the pin1 auxin carrier. So now we understand the dynamics of the pattern, and we understand how the pattern is created. What can we do about changing the phyllotaxis of a plant? And I'll just show one example here. What we have here is a view of 3 different plants of different genotypes, different mutations. And we've looked at them because of a specific prediction of the mathematical model. Now you saw the differential equations, and you can see that each of those differential equations had a bunch of letters, which are the parameters for the models. And we parameterize the models using, for the most part, literature values for the diffusion of auxin, the rate at which auxin moves through membranes, the rate at which pin1 can move auxin through membranes, and so on. And then we tested the model to see which of the parameter changes would change the phylotactic pattern. So we changed the amount of auxin, didn't have very much effect. We changed the rate at which things moved, it didn't have very much effect. But one thing that did have a strong effect was the size of the meristem and the relative size of the meristem to the size of the region in the center of the meristem where primordia do not form. So that a bigger central region where the primordia don't form and a bigger periphery led to changes in the phylotactic pattern in our computational model. So we went and took a series of mutants that have different phylotactic patterns with these various names, and we went in and we looked at where the cells are that are in the central region of the meristem that divide slowly and won't make primordia, and how big the overall meristem is. And we saw that those varied in these different mutants. And as a result of that variation, when we look at the mutant flower stalks, we look at the fruits that sit around the stalk and their angles, and you can see a whole variety of different angles here. The result from changing the relative size of the central region of the meristem to the size of the meristem as a whole. So now all we have to learn how to do is change the relative size of the central zone of the meristem and the meristem itself, and we do know how to do those things. And as a consequence, we have control over the phylotactic pattern and can begin to think about changing it in agricultural settings. So with that, what do we have? We have a molecular solution to the old problem of the origin of phylotactic pattern. We have a new class of pattern formation for developmental biologists. A new class of model of pattern formation is that arises from cell-cell communication, because we have what a developmental biologist would call a morphogen that moves from one place to another in an organism and at high concentration induces the formation of some developmental phenomenon. In this case, the morphogen is auxin, And the developmental phenomenon is the formation of a new flower. And we have a regulated transport model of morphogen movement rather than the reaction diffusion models that Turing invented to explain phylotactic pattern, but that don't, or the lateral inhibition models, as Drosophila or Caenorhabditis biologists have discovered with Notch-delta interactions, which create pattern. This is a third class of developmental model to create pattern. We have a demonstration that local communication between neighboring cells can give rise to global patterns. You'll notice that there is nothing in our model that had a cell knowing anything other than its own concentration and the concentration of auxin in its nearest neighbors. And that completely local interactions create the global phylotactic pattern, which is a lesson for all of developmental biology. And then we have an approach to understanding the path from genes to phenotype by explicitly considering cells and gene action and regulation of gene action and movement of chemicals between the cells, something that we can observe using these live imaging methods that we and other labs have developed over the years. And all of that gives us the ability to alter plant architecture in a predictive manner so that now we can go back and change the phylotactic pattern in plants as we wish. So what conclusions can we make about the potential agricultural use of this? It's a long way off, that's one conclusion, because we're just now learning how to do these things. But present-day agriculture depends on methods that were developed in laboratories 10 to 100 years ago. The future agriculture will depend on the basic science that we're doing today and that other plant research laboratories are doing today. And so by combining experimental and computational approaches, we can project to a future where we have predictive models of plant growth and of plant development and can generate the plants that are necessary for human welfare. So when should we do this? We have to do it now. And so now is the time to start understanding in this sort of computational depth how plants grow and develop, and in the near future to start applying this to human needs and to the improvement of the human condition. I'd like finally to thank in this long slide some of the many people who participated in this work recently and in the slightly more distant past, and this is by no means the full list of the people who've been in my lab and in the labs of our collaborators. And one final lesson I want to bring to this part too is that we did not do this work alone. It is a collaboration of a number of different laboratories with a number of different talents. We're the microscopists, and we know things about plant genetics, but the mathematics and the computational modeling was all done in collaboration with mathematicians and theoretical physicists at a variety of different institutions all throughout the world. And without that sort of collaboration, we would never have achieved this small level of understanding that we have now of how plants grow and how plants develop.